Today I will talk to you about chromatin. So it is.
Yes. Yes. With this.
Okay. Okay. So the goal of the study is to model the DNA replication in the three-dimensional space. And particularly, we are interested to study some structural features of chromatin. And even more interesting, we would like to model chromatin during replication stress conditions. So speaking about replication, there is a particular process behind it. There are some specific losses of chromatin which are capable to initiate DNA replication, and these are called replication origins. Typically, we can define a probability mathematically that in a specific place in DNA and a specific time, there is a probability that the origin may fire or not. And in this way, we can make a Monte Carlo process where we make like a kind of simulation that in each step we check if a random number is lower than this probability, and if it is lower, then the origin can initiate replication, and then the forks propagate bidirectionally. When a fork meets another one fork, then we have the two forks merge, and typically the replication ends when all replication bubbles have merged. So usually all the previous studies were studying replication in this way because only one-dimensionally, using replication timing curve data to define let's say this kind of probability. This was not trivial, and this was the main reason why they were fighting a lot with this. Also, there are quite a lot of cases about how this replication can happen because there is homogeneous, inhomogeneous replication. But there are not a lot of studies that they model the 3D structure, and especially the 3D structure with the interplay with other proteins. Yes. So my idea is to create a graph, and in this graph I have node states and links. And what I'm doing is that I'm trying to make a Monte Carlo process where in each step we can either propose a node change or a link change. Now, what is a node and link? A node change represents a epigenetic state. It is like abstract connection with the fiber subcompartments that exist in chromatin. There are also edges. The edges of the graph represent loop extrusion factors. So they are SMC complexes that they make loop extrusion. And also backbone. So the main idea is that we use a Hamiltonian to process all this. This is also something very important because the Hamiltonian formulations help us to create models that are scalable. So if we want to include another one biological process, we can just add a new term in the Hamiltonian. So this is the outline of Replisage. We import replication timing data or 3C type experiments. So there are these two types of data that we need. From replication timing data, first we run 1D replication simulation, and then we have, let's say, another one model, which is not exactly Ising model, but let's say this is the idea that I have a network, I have a POTS model to model the node and link, the node states, and for link states I have written my own Hamiltonian. And I iterate this according to some biophysical assumptions that I will talk now. And I create a set of states. Having this set of states, an ensemble of states, I can create an ensemble of structures. And from this ensemble of structures, I can make some validation of it. which is not so easy. So in general, in this simulation, there are three phases, like in the cell cycle: G1 phase, S and G2M phase. In G1 phase, we have actually loop extrusion. We have so the SMC complex that loads the DNA fiber, it extrudes the loop, and typically it stops when we have a CTCF complex. And this all is modeled with a Monte Carlo energy that I will not show you now because I want to be fast. And therefore, this was like my first work that I did with this kind of formulation, only with loop extrusion. That I had, let's say, only the 3C type experiments, and then I created some kind of ensembles of possible configurations of loops. And from this, I created the 3D structures. I averaged them, and then I had the average heat map, and I tried to reconstruct it. This is something that a lot of people try to do. But I did it for first time using a Monte Carlo energy. And this was one of the reasons why I was able to make some steps further. So for example, when we have the S phase, we have the replication, and then we can apply some some rules that we know from papers. For example, we know that the replication forks acts as barriers for cohesins and SMC complexes. Yes, and another one important thing is that replication is much slower than the loop extrusion. So this helps us to have two time scales. Like the replication is almost, let's say frozen if we compare the time with loop extrusion. The S phase lasts for much longer time. And of course, in G2M phase, we have there are some recent works that they speak about it. Actually, of Job Decker, that they are trying to model like two populations of SMC complexes, cohesins, condensins, and then they show that condensins extrude longer loops, and the two chains can separate from each other. So these are some of my results. This is how it looks. In S phase, usually we have decondensations, not usually, always, because of the replication forks are pushing towards each other the two chains. And in G2M phase, we usually have further compaction because of the condensins. And for example, if we calculate the average loop size, we can see that there is a decrease in S phase. This is something that we have also validated with single cell high C data. That something it really happens. And another one important thing is that during replication stress, I model replication stress by activating more origins more often. Then we have smaller loop size. This is something that we have also validated with high chip experiments with replication stress conditions. And this is something also important, that there is some very standard pattern in the gyration radius and the convex hull volume. And we have reproduced this by hard coding these 3D structures from single cell experiments. And we see exactly the same pattern. So it means that here with much less data, we can reproduce a very similar process only by doing some Monte Carlo. And I tried also to run it for all chromosomes. I verified that these results are consistent, so of course for low resolution models. These are the validation, at least this is the qualitative validation, because I also have quantitative validation with compartments and comparisons with heat maps. But I think that this is the most important one. At right, I have the high chip data that we see that there is a shift in the distribution for lower loop lengths in case of replication stress. And with single cell data, we observe similar patterns, that in S phase we have smaller loops. And this is my work. So just to summarize, we have succeeded to have a model that models the whole cell cycle with a minimal, let's say, input data set. We use, let's say, some observations, some biophysical laws that we know from bibliography, and we are reconstructing some other ones that we know independently. And so we have some validation methods, both qualitative and quantitative. And of course, one very big advantage that I think is that we can model replication stress, and that this model, it is very easy to modify it if you know exactly what you want to model, because you can just add or remove some terms in the energy very easily. So that's it. Thank you very much.
Thank you. We have time for a few questions.
Thank you very much. Very interesting. I probably missed it. How do you model the effect of topoisomerases? Basically, how you avoid knots?
Yes. So normally, yeah. So this is something that I do. Actually, I model topoisomerase. The knots are avoided anyway because of the excluded volume potential in the 3D structure. But I model topoisomerase because it is needed for the separation of the two chains at the end of replication in G2M phase. So how I do it is that I have, let's say, a soft and hard excluded volume potential. And as a Monte Carlo process, let's say I have some very few losses of chromatin that are they have this very soft potential of excluded volume.
So the chains can cross.
Yes, they can cross. So this process can or create knots, or they can destroy the knots. But usually when it is very small amount of this topoisomerases, which is how it is in reality, then we avoid the knots like that. And the chains are more easy to separate.
Any other questions? Thank you, really cool talk.
Thank you.
I just wanted to ask about, I know that the origin mapping is an active challenge in the field of replication.
Mhm.
So given your simulations, how did you approach validating that aspect, especially when you say that the replication timing curves, you were able to replicate them? Did you compare your earliest local replication loci to existing datasets from small datastructs?
Yeah, so what we do is that we make, let's say, we define from the replication timing curves, we define some functional, which is called initiation rate, and it is function of place, space, and time. Yes, so so in this situation, this defines the Monte Carlo probability that an origin fires. So I can run, let's say, some hundreds or thousands of simulations, and then I can see how the replication forks propagate, and I compute some statistical average of it, and I can compare with the actual data that we have. And actually, this is not a very difficult task to do. We can achieve even 100% accuracy on this correlation. Yeah. So yes, I have done it. It is quite easy to do it, but it depends. Let's say how much you care about it, to not assume too much about it, because how to say that, you can always just assume that the origin fires like in the time very close to what you observe, like in the experimental data. But in this situation, for this simulation, we care more about the interplay. So I'm okay that I just have something that works. So thanks.
Okay, just a very, very brief last question.
Mhm.
all the work you presented is on the normal genome, right? Have you tried running some simulation where you have the chromosome deletions or translocations?
So I have, for now, I have tried to run it for 2 datasets, 2 cell lines. The one is GM 178, 2778, and the other one is K562. So it is a cancer cell line. For now, it is probably working in the first stages, yes, and we focused on the validation, but I have tried to run it for these 2 cell lines. You can easily run it for any other data.
Thank you.
Thank you.